I am a medical student at Oxford. I'm a fifth year medical student, but I took 4 years out from my medical degree to do a PhD or a DPhil. So Oxford calls it a DPhil because they just do. And I specialised in the genetics of a rare lung condition during that, and I worked with some about gene editing, kind of from a bit of a historical perspective, just so that you understand the tools that scientists have access to. It's a topic that I find really interesting and exciting, and that's been reflected by the awards that this kind of science has been winning lately. So it's a really lovely opportunity to be able to talk to all of you about it. I'm certainly expecting lots of questions throughout the talk, so please do pop them in the chat and I will keep an eye on it as well as going through this. I have 2 question breaks in my slides as well as time for questions at the end. So if I don't get to your questions like just during the talk, then I will deal with them when we get to the, free to ask them when we get to the question slides. So we're starting off with a little warm-up quiz. So just grab a pen and paper and then write down the answers to these 6 questions. And then the 7th one, you'll need to see the genetic tree on the next slide. So starting with how many chromosomes does a human skin cell have? I'm hoping, depending on where you are in your A-level studies, hopefully this knowledge is not too far in the depths of your brain. What proportion of our DNA is shared with bananas? Drosophila is the name of which commonly used experimental organism, and I may be showing my age with Gregor Mendel because this was a key part of the GCSE biology syllabus when I was 16. Was knowing who he was. Then dystrophin, which condition is that linked with? Hazard a guess at the number of Nobel Prizes across the 3 sciences that have been won by women. And then I'm going to call for answers in the chat and hopefully you guys will join in. So how many chromosomes does a human skin cell have? Any suggestions? So Remy, 46, 23 pairs. Nice, thank you. Right, what proportion of our DNA is shared with bananas? 50%. 50. Yeah, so basically, yeah, we're not too far away from a banana is essentially the answer there, but far enough away that we don't tend to use them as a genetic model. What is a Drosophila? Does anyone know what a Drosophila is? There's nearly, no, so it's not a mouse, it's a fruit fly, and you're going to see some pictures of them on the next, in a couple of slides time. Does anyone know what Gregor Mendel did? Either his like day job or what he did in science. Any guesses? He was a biologist. The pea thing. Okay, yes. Thank you, Kira. I knew that someone would be like, yeah, he bred peas. He was a monk and he bred peas and worked out like inheritance of basic dominant and recessive genes. Fantastic. I like you guys already. Right, dystrophin. Do we know which X-linked condition that's associated with? This one is quite hard. I had to put in some hard ones because, you know, we can't have them all as like, how many chromosomes does a human skin cell have? So dystrophin, I think some of you may kick yourselves. Not Down syndrome, so it's associated with Duchenne's muscular dystrophy. I'm just going to type that in the chat just in case anyone wants to make a note and look it up later. You will almost certainly, like, it will ring bells when you look it up. You'll have heard of it in some shape or form. And that's the name of the gene. So dystrophin, dystrophy does kind of make sense. Right, best guesses out of 357 Nobel Prizes across the 3 scientific disciplines. How many female winners have there been? 4. Okay, so it's not that bad, but it's not that much more than 4. So 80, it's not that good. So between those 2 numbers, 20, okay, so it's 22. So not fantastic, and it is rapidly changing, but just thought I'd include that because 2 of those winners out of 22 are coming up later in the talk. And now, if you're a slick geneticist, you may recognize some of the names on here. What condition is shown being inherited in this family tree? And bonus points or haemophilia. Fantastic, Mariam. And do you know how, like, what kind of inheritance is it? Is it recessive? Is it dominant? Is it X-linked? it's X-linked, and it is recessive, but it's X-linked, and we know it's like recessive because the women are light pink, so they are carriers because they have 2 X chromosomes. The only affected people are men. Okay, so they're the dark red ones, and that's because they only have 1 X chromosome. They don't have a fallback gene to work, so they wind up getting it. And this is basically an advert for don't marry your cousins, because you'll increase your likelihood of getting a recessive condition. And haemophilia is one of the things that ran in the European royal families when this was like a big thing. So what is genetics? So this is what I'm talking about today, but I want your guys' thoughts. Like, what is genetics? What do geneticists do? So ideas in the chat, please. If I say I'm a geneticist, what does that mean to you? What defines you? Oh, that's nice. I like that. Map out genomes. Okay, interesting. Do you want to tell me what's in a genome? And I know that there are more than 2 of you in the chat, so put down your ideas. All DNA, yeah, all DNA is in your genome. I have a little pet peeve about people calling things junk DNA. Work with genes and genetic diseases, genetic engineering, fantastic. Okay, so we are getting some answers now. Great. So how do you think we can investigate the function of a gene within the genome? So people have talked about genomes being all of your sequence DNA. Have you read, heard, seen anything in the news? How could you investigate things? Like if I wanted to investigate that dystrophin gene through switching it on and off, fab. So I could switch it off and see what happens. Or I could maybe try and take something with a mutant and then reinsert the gene and see if that like fixes the problem. Do you have any other ideas of how you could investigate a gene within the genome? So you guys are all A-level students, I believe. So obviously genes are encoded in DNA, but what are the kind of downstream things from that, the downstream products of that DNA that could be affected that we could also play around with as geneticists? RNA, fantastic. So we could knock out the gene, but then reinsert RNA. Twin studies are a really interesting way to study genetics on a population level. Introns and exons, really, really important to know about those. They're used in the construction of some of the things that we use to muddle around with DNA. Proteins, yeah, so those are the big ones. So you can come in at DNA, RNA, or protein level, and you can investigate the levels of DNA, RNA, and protein. Obviously, DNA levels aren't really going to change because we know that your cellular DNA, even though it changes when your cells are replicating, if gene expression changes, does your cellular DNA change? Think about that. So if gene expression changes in your like cell culture in a dish, does the level of DNA in those cells change? So gene expression is changing. Does the level of DNA change? What would it depend on? Tell me. If you're going to say it depends, I'm not saying that that's right or wrong, but I want to know what it depends on. Any ideas on what the gene codes for? Okay, so I think that you could technically be right. I'm not sure if you know why you could technically be right. So if I have like a little petri dish of cells and I've turned on a gene, so that gene is expressing more, what I will see more of is its RNA and the protein downstream. Okay, so we know that that DNA is not then going to replicate and give itself like 5 copies of the DNA. But say I was turning on a gene that then turned on the cell cycle, so I got more cellular replication, then if you were talking about the whole petri dish, then I may see more of that DNA, but I'd see more of all of the DNA of the cells. Does that make sense? Yes, thank you. We love the feedback. Okay, so let's start at the beginning. We've talked about Mendel and his pea plants. This was actually really, really important and like seminal scientific work. And when people say that, they mean that it was like a pivotal point in science. So we didn't realize it at the time because he was a monk and he was working with peas and he couldn't explain how he could predict that like 1 in 4 of his pea plants would be short peas or tall peas or whatever. But it was really, really important. And that's where all of these like Punnett squares that you guys learned to draw came from. Now, what is the difference between these 2 flies in the top right corner of the screen? This is the more obvious spot the difference question. What is the difference here? Red eye and white eye, fantastic. So The other thing that happened early on in genetics, this is like early 1900s, was that people learned that you could introduce random mutations in whatever was coding for inherited traits. So at that point, we didn't know what DNA was. This is like the 1910s, but someone worked out that if you sat fruit flies around like particular substances, you could introduce mutations and then you could breed them and look at the transmission of these characteristics through offspring. And fruit flies are useful for this because they breed so quickly, basically. So they're a really, really useful genetic tool. If you want to look at successive generations, you don't want something with like a 30-year generation time. What's the difference between these two flies at the bottom? One is hairier than the other. I mean, this is essentially true, right? One of them has these massive eyebrows and the other one doesn't. I mean, they are above the eyes. I don't think that eyebrow is a technical fly anatomy term, but they essentially have eyebrows and don't have the other thing. Isn't something on the outside. So it's these eyebrows things that is the difference. So what the other big thing that came about with fly genetics was that people worked out in about the 1940s that radioactivity had this mutagenic effect. So mutagenic, creating mutations. So it's creating mutations randomly in the genome. We can't control what gene gets knocked out, but you get these weird mutant flies that arise, some of them that have extra eyebrows, some of them that would have like bonus wings. So there were gene knockouts, but this was still really before people knew what the heritable information was, before we knew what DNA was. This is all way before 1963 structure of DNA being discovered. So these basic gene knockouts are pretty much the Duplo of gene editing. It is still really cool science. Like you look back and see what they were doing, especially before knowing what DNA was, and that's still really interesting. And then these guys had a massive fruit fly lab and actually won a Nobel Prize on this. They were particularly looking at genes involved in early embryonic development, most of which are the same between fruit flies and humans. So these are the genes that, like, you know, you have to go from a ball of cells into an adult that has a head and feet. And obviously there's difference between your head and your feet, but how does the embryo even know? Hox genes, fantastic, Clara. So I'm doing a layperson summary of Hox genes, but Clara already knows what I'm talking about. So these genes turn on segmentally as you go down your larva or your human embryo, and they say, oh, this is the point at which your arms need to grow out. So if you duplicate that in a fruit fly, then they get another set of wings. And those genes are, you know, conserved across human development and mammalian development, but they were all discovered in the fly. And the fly is so important that its genome was one of the first to be sequenced, and you can like Google the genes basically on Flybase. So question pause if anyone has any questions to ask now, and then we're going to do a bit of a deeper dive into gene editing as opposed to gene knockouts, including going over CRISPR. So any questions? I was told you guys would ask loads of questions, but Rem, you might just be wrong.
So, I mean, they have all the answers today.
How dangerous is it messing with genes? Oh, okay, so fun question. Okay, so in theory, it could be very dangerous. So the most dangerous thing, realistically, is something called off-target effects. So normally when we mess with genes now, I mean, we're still essentially messing with genes. Normally when we mess with genes now, we're like, okay, so I know that I want to target, for example, the dystrophin gene, and I want to introduce a change in it, and I don't want to introduce that change in any other gene. And the difficulty is that you might introduce a change in another gene, not realize that you've introduced a change, and then like 10 years down the line, everyone that you've gene edited gets leukemia and dies. Bad. Right? So the concept of off-target effects is probably the thing that people are most worried about as a danger to like organisms that we edit genes for. What do you do on a normal day as a geneticist? So obviously I'm a medical student now, but I did really, really like being a geneticist, or so I was essentially a molecular biologist. Is the kind of science that I did. So that's things like cloning DNA, making like rings of DNA and putting them in cell culture. And then you could use that to make loads of protein, and then you could do experiments on the protein. And I would spend time sometimes like harvesting tissue from mice, sectioning it, staining it different colors. I did quite a lot of live imaging using like high resolution or laser microscopes and fluorescence. What else did I do? I did quite a lot of staining of different tissues and embryos to look at gene expression and then look at protein expression, because as you guys know, if you want to look at gene expression, you're going to look at the RNA, but then the protein expression could be different to that. So you have to double check and do both. So I had experiments in like adult animal tissue, embryo experiments, and then experiments in cell culture, which were all in mouse-based systems. I collaborated with scientists that were working on the same thing, but in zebrafish. I would prepare talks, sometimes like this, but often for conferences, or prepare papers or posters. So really, really mixed. I liked being able to be in charge of my own time, and it was really fun. Like I still really, really enjoyed being at the lab and essentially like messing about with genes, as Abilene says. Okay, the questions are coming through. I'm gonna answer a couple more. How much is applicable from Drosophila to humans? So if you think about it, lots of the cellular biology stuff is the same, okay? So it's not like they have a whole different way of doing respiration. And I'm not talking about breathing, I'm talking about like, you know, the cellular process. So all of that stuff is the same. And lots of the actual protein level biology is very, very similar. The early embryo stuff tends to be more similar than the later things, but, you know, flies have eyes, right? So some of the light receptor proteins will be the same too. So in terms of heritability, there's a lot more than people thought at the first time those experiments were being done. I will say that lots of the, you know, we don't know everything about everything, but we do know quite a lot about the Drosophila genome now. How ethical is it to employ someone based on their genome? I'd say currently incredibly unethical. In the future, probably still incredibly unethical. There's so much that we don't understand about the genome that just, I think that would make no sense anyway. How do you choose which genes to remove or add when doing genetic engineering? Normally you have like, it's like all kind of science, or you might have done it in maths where you do like H0, this is my hypothesis. So you maybe have a gene of interest. The one that I was interested in my PhD, there were big screens of the mouse genome where they were going through and knocking out each gene individually and seeing what the effect was, because the vast majority of genes, we don't know what they do still. People are sometimes surprised by that, but we don't know what they do. So they were doing that and then seeing if it had an effect. And someone did that to the gene that I worked on, and they were like, oh, it seems to have these particular defects in the adult. We don't know anything else about it. And then I stepped in and was like, let's find out what it's doing in the cells. How do you identify how long or what the gene is due to the long and repetitive nature of the DNA sequence? So genes have like fairly readily identifiable start signals and end signals. So computers are quite good at identifying that now. In theory, could you alter genes? We're coming back to that, Adriana, so I'm not gonna, and if I say your name wrong, I'm really sorry, guys. Do you feel that excessive gene editing could endanger natural selection if it's out of control? I think that there's the possibility for that, not just for natural selection, but for the excessive gene editing could be out of control. And we're going to talk about the ethics of it later on in the talk, so we'll move on there. Just checking the time. Okay, so how do we gene edit? Yuan, I will come back to you at the next question break, if that's okay. Right, I'm turning the chat out of my screen so that I don't get distracted by you guys in the next like 10 minutes. So moving on to the mammals, key things that you need to be aware of are homologous recombination and then site-specific nuclease activity. Now, I said I was going to turn the chat off, I'm not at all. Nucleases. You guys know about enzymes. What do you think a nuclease might do? What does it digest? Ideas in the chat. Nucleotides, fantastic, correct. It cuts nucleotides. So site-specific nuclease activity may sound like a complex title, but actually we're just talking about site-specific, so particular bit of DNA, nuclease, enzyme that cuts DNA activity. So scissors that cut DNA where that you want them to. Right, homologous recombination is the first thing that you guys need to know about, and this is like the we always use this as geneticists. This is like the go-to. Almost all of the things that I'm then going to talk about after this come back to HR. So you know that you have 2 copies of all of your genes. I know that you guys know that. Is that how they cut the insulin gene out with site-specific? With restriction endonucleases, I think is how they originally cut it out. But that's going back into like 1980s science. And I'm pretty sure I read the paper at one time, but I'm not going to lie, haven't read it for a while. So that may be a job for you to Google yourself as well. Right. homologous recombination, before I get fully sidetracked. So we know that DNA, you have 2 copies of all your genes. You guys know that. You told me about the number of chromosomes at the start. So if you have a double strand break created by some kind of exogenous things, this might be like an oxygen free radical, it might be radiation, it might be other chemicals, particularly chemotherapy drugs tend to do this, then the missing information is provided by that second copy of the gene, and that's used as a template to restore the genetic information. And I've included this example diagram because I'm guessing that someone amongst your number may well know which condition BRCA1 and BRCA2 are related to. Answers in the chat, please. If you know what condition BRCA1 and BRCA2 are linked to. Breast cancer, fantastic, and also ovarian cancer, okay? So is it that surprising then that these proteins that are involved in DNA repair are involved in cancer? I mean, no, actually, not really, because we know that cancer cells have really distorted and warped DNA, and actually having a propensity to DNA damage is one of the hallmarks of cancer. So that's just a little extra tidbit. But basically, the cell uses the other strand of DNA that is there as a template. That then means if we supply it from outside the cell, like if we make a strand of DNA for it to use as a repair, it will copy our information into its genome that we've given it. So basic diagram of how this works. These are cells from the inner cell mass of an embryo. Okay, so these dark blue ones will go on to become the embryo. The light blue ones go on to become the placenta, just for those of you who are interested. This is our little genetic cassette that we're going to introduce in. So a genetic cassette just means a structure of DNA that you've made and planned. So like I would literally use a computer program to design this and be like, this yellow bit at the end matches up with this start of my gene of interest, and then the other one matches up with the other ends. Those are complementary sequences. This neo-R bit is a neomycin resistance cassette. So what I'm going to do is I'm going to electroporate these cells, so give them an electric shock to introduce my little genetic cassette. And then homologous recombination will take place. Most of those cells will fix their DNA using their own DNA. Like they won't use my little sneaky genetic cassette, but a couple of them will. And then what I'm going to do is pour, I mean, obviously it's more technical than this, but I'm going to pour neomycin all over these cells. And if they don't have my sneaky genetic cassette introduced into them, they will die. So we wind up just with a population of these red cells that have all taken up my cassette. So what I've done is I've replaced the gene of interest with my genetic cassette that no longer has the gene in. So any organism that winds up with all of its DNA comprising of this is going to not have that gene expressed. So nearly there, but some of you will realize that, you know, we're going to inject 2 into this. That's not the whole organism. So what happens? Well, we inject that into these blastocysts. They go on to become every single cell in the mouse's body. So you get a chimera, and we know that it's worked because, and this is literally what they do, because we make those red cells from a brown mouse and we inject them into a white mouse. So if it's worked, you get a mouse that is chimeric. So it's got brown bits and white bits. And from then we breed them out. So you breed them with normal mice. And because all of the cells in the body are a mix of these pink ones and the yellow in this slide or whatever, then that means some of the germ cells, so some of the eggs and some of the sperm, are also a mix. And we know that, you know, that egg then becomes 50% of the offspring's DNA. So you've then got a heterozygote knockout of the gene that you were trying to knock out. You can breed those together and create homozygous knockouts. Does this all make sense? Because it is a really complex topic. Is this selective breeding? You select the cells and then you breed out the chimera that you've created, but it's not selective breeding in terms of like, I want to make this cow beefier, so I'm only going to pick the beefy cows and mate them together, or the beef cow, beefy bull. What happens if the new cells are rejected? They're not, because they're introduced into the embryo. Okay, so this mouse is literally a mix of cells that are the yellow ones and the red ones. Obviously in the adult mouse, in this cartoon, we don't see it, but here in this picture here, you do see that it's a mix of the brown cells and the white, like the red cells and the yellow cells, because this mouse is literally like its fur is different color. And this mouse is a lab mouse, so it should be like very genetically homogeneous. Its genes should all be the same. It should not be different colors. It should be either brown or white or black or whatever the line of mouse is. Do they have to be a lot? Yes, they do. You breed this out a lot, and then you have to breed your targeted mice together, your heterozygotes together. So they're very inbred. That's the other thing. So that's not so realistic. And you need a lot of mice to maintain a colony. And the generation time of mice is like three months. Do you recommend any sources? I will look some up, Latoya. What happens if the gene is turned off at the start? What do you mean by that? Do you mean like you completely inactivate it for the organism the whole way through? Right, I'm going to move on to the next slide because you said dealing with sexually. Do you think it was ethically correct to do this? Okay, so we are going to do ethics later. So I'm not like batting a question down the road. I'm batting it like 5 minutes down the road, but we'll talk about it in a minute. Right. So that basically is how we would make a gene knockout that had been happening for like the past 30 odd years. Okay, if it accidentally turns off, sometimes happens, you do get random mutations even in an experiment like that, and could make your experiment useless. If you have enough mice, you can probably deal with it. If you have enough heterozygotes and stuff to recreate your colony. Right. The next big step in the science, like the history of gene editing, was the introduction of these enzymes. So they are proteins. I know that you guys know what enzymes are, but I have to make this clear, that have a site that is specific for a DNA sequence and has a cleavage site. Now, because this is protein binding to the DNA, and we know the whole like lock and key induced fit enzyme hypothesis thing, you need a new protein for each new site of DNA that you want to target. Now, our computer prediction tools are fairly decent at working out what amino acid sequence you're going to need to get the right shape to bind to the DNA, but they're not fantastic. So you need new enzymes for each gene you want to target, and essentially it's a massive faff. They don't tend to be very, sometimes you get ones that work really, really well. They're really specific and they're fantastic. Lots of the times they have off-target effects like we were talking about earlier. And they're actually like a pain in the bum and they're a real struggle to make. Talons, same thing. So a section of protein that binds to DNA and introduces a break, sometimes really good, sometimes a pain, very expensive. It takes a long time to engineer the new enzymes each time you want to change a different gene. CRISPR, this is like my favorite topic. So excuse my like really drippy slides here. So I used to give this talk or like a similar talk to this and be like, you just mark my words, this will win a Nobel Prize. And then it actually did in September. And I was so happy. So these two women are like my heroes. Gosh, I'm really sorry that I'm embarrassing myself like this, you guys, but I hope that one day you find something that you love as much as I love this stuff. So Jennifer Doudna is on the right, the left even, and Emmanuel Charpentier is on the right. They discovered CRISPR-Cas9, which is what I'm about to talk to you about. And I actually saw Emmanuel Charpentier give a talk in Oxford about 5 years ago. And it was so like, even then, you know, their paper was only published like 9-10 years ago. And even then it was such a huge thing in the field and everyone knew it was so big that like people were sat on the staircases trying to get into the talk. Like it was so popular and it was really cool. So how does this system work? The main difference here is that you only like what you use to take your DNA scissors to your DNA, so this is an enzyme, Cas9 enzyme, is a guide RNA. And every time you want to target a different part of DNA, you just need a new guide RNA. Now, obviously, like you guys haven't been to the online science supermarket, but I have. And these are quite cheap, like between 25 and 100 quid maybe to get a section of DNA that's like 20 to 20-ish nucleotides long, okay? So cheap, and easy as well, because I would literally just be like, I would like 3 C's, 2 G's, a TA, TA, G, C, C, C. And then I would have that arrive like at the lab in the post the next day. Okay, so this might sound a bit weird because I know that you learn it in school and that seems very removed from like someone in a lab on their laptop ordering some DNA, but that's literally what we do. So they're cheap, they're easy, and even just bringing in a double strand break is really, really cheap and easy to do. And it's incredibly, incredibly precise. Where does this DNA come from? There are companies that make it. There's like 4 or 5 different ones. So, and they just make it like they make chemicals. So artificially made, yeah. but it's the exact same like chemicals that you have in you. Like at the end of the day, we are all like a soup of chemicals, right? So if we want to be that reductionist, but yeah, you basically order it, and sometimes there are problems, like you start using it and you're like, nah, there is no way that this is the right sequence, it's not working. And because it's quite cheap, sometimes you'll be like, it must be the DNA, it's not my science, so I'll just reorder the same DNA. And sometimes that is the case. Is the guide RNA complementary to the target? Yes. And the good thing about us knowing like the sequence of lots of genomes and stuff now is you can literally pull up a genome library and be like, okay, so this is the sequence I want to target. This is the complementary like guide RNA sequence I'm going to use. Does it bind anywhere else in the genome? And you can search your genome, like your base pair sequence, and see. And obviously, if it does bind somewhere else, you don't use it. You edit it a bit more and make sure that it's not going to bind anywhere else so you don't get off-target effects. What does price have to do with DNA? Well, because science costs money. And if you want to do science experiments, you need a grant. Someone has to pay for that. If I want to knock out the gene that I worked on, then if I can do that in a couple of months with training from freely available papers on the internet and resources that cost a couple of £100, my supervisor will probably fund it. If it's going to cost tens of thousands of pounds and take a couple of years to get a specific like zinc finger nuclease, probably not going to happen. Okay, so sadly, money does make the world go round. And the big reason this revolutionized science is that it's so cheap and easy that anyone with a standard molecular biology lab could pick it up and then use those techniques. What costs that much money though? Is it the technology? So with the other mice are the one thing. Animals cost a lot of money. So the old style, let's like edit the stem cells and then breed them out. That's multiple generations of mice. So it costs a lot of money to house the mice, to look after them, and to have people that are paid and trained sufficiently to look after them. The enzymes, like lots of the time, sometimes universities have their own in-house teams that will like design your enzymes and stuff for you, but often you would have to contract that out to a private company, and that costs a lot of money. And like I said, sometimes that doesn't even work. So then you're left in a situation where you've spent like 10 grand and you've got nowhere. Okay, so money is important in science. So as well as doing knock-ins and knockouts, you can do loads more with CRISPR by tweaking the enzymes. So we can introduce a really, really specific double strand break, which will half the time lead to a knockout. But we can also repress the gene, activate it, or even edit the epigenetics. Do you guys know about epigenetics? Yeses or no's in the chat would be helpful. No, okay. Yes. We're like, okay, so some people are saying they do know a bit. Some people are being very helpfully honest and saying not really. Some people don't know anything. So epigenetics is like the wide scale on or off of lots of genes across your genome. So your body can basically like turn up the volume and turn on a set of genes that, for example, are to do with your metabolism. Say if you were in a famine and it needed those genes to be on. And there are particular marks on your genes and on your genome that send a signal to the enzymes involved in that, being like, hey, turn me on, please. And there'll be like hundreds of them potentially that get turned on. So we can not only just turn these genes off, but we can edit those signals. So you can look at like the much broader scale way that genes are manipulated just normally physiologically in humans, which is really helpful. Because if you think about it, a full-on knockout mutation normally causes a lot of disease, right? So it's often lethal, but if it's not lethal, often like reproduction is reduced or it's life-shortening. So they're quite rare and they're quite severe. But most disease in the world is caused by or is influenced by genetic effects that aren't as strong, but they're still there. So, you know, lots of little genetic effects that altogether have a strong impact. And like, we know that genetics influences heart disease. We know genetics influences blood pressure. We just don't fully know how. And that's pretty much mostly because it's lots of little effects potentially mediated by these epigenetic things, as opposed to just like, bam, one big gene knockout. So this was a question pause. And I know that there were some ethics questions, which I'm going to back to the next slide because I was going to ask you about ethics anyway. So I'm just seeing if there are any more questions. What genetic process is used in IVF that genetically modifies babies' features, e.g. the colour of the eyes? All the ones I've talked to you about, potentially. Do you think CRISPR could be used to treat and prevent cancer? Potentially. Right, we're going to move on, and I'll just take questions as they come, because I know that we're running out of time and I don't want to run out of time. So, key applications of CRISPR-Cas9. There will be loads more of these, but these are the basic ones. So really useful in research. You can knock out genes, introduce new code. It's a really, really specific and easy way of playing with these things. I know that it may seem, when you're sat there studying it, like an experiment not working isn't really a thing, but believe you me, if you've sat there and done the same experiment time and time again and it doesn't work because your tools are not very good, then something like this is revolutionary in terms of what science can actually churn out and how productive it is. Can visualize gene expression. Do you think, oh gosh, yeah, I mean, that wouldn't work at, like, there are so many scientific reasons why that would do nothing to your cells, this like injecting you with Cas9. Like the immune system would be on that protein like a shot, like it was a stunt, is what I'm saying. Switching genes on or off, RNA editing, treating genetic disease, and potentially treating infections. This could be a really interesting avenue for antibiotic resistance. This was originally a biological defense system. I 100% recommend Jennifer Doudna's book, which is called A Crack in Creation. Really, really interesting story of like how they developed CRISPR-Cas9, because this is 2 labs, one working in France, one in the US. So there's like a lot of communication and science sharing that has to go on for that to work. Big ethical issues with gene editing. I know this is in the syllabus. Put your ethics questions in the chat now. I know someone said, did you think that using animals was ethical? Mice, I could just about square myself with, especially when you then like see patient experiences of these conditions. Like the kids with the lung condition that I worked on could wind up needing lung transplants, and it's quite rare. So just no one really works on it. We don't understand all the genes that cause it. And in my mind, I could square that with what I did. But I know that some people can't, and it's not something that sat easily with me. You make every effort as an animal researcher to make sure that you use the smallest amount of animals possible, and you do not let them suffer and all of those things. Like my name was on those cages, and if the mice were treated inappropriately or like had, you know, I would have protocols in place. So like if blood was taken, and if like 1 more mil of blood was taken than was allowed, I would go to jail because of that, because I was responsible for those animals. Like I had a license for them. So in the UK, I would say we are very good at regulating animal research, and we are very, very good at making sure that it's not done in ways that introduce more harm than is necessary. I would say other countries maybe not so much, and that's maybe where people really need to think about things. So in the UK, humans are excluded from the animals in science legislation because they're different. But in the UK, all mammals plus all squid, because squids are very, actually very sentient beings. If you like squid rings, this is maybe not a good day for you. All of those animals are under the Animals in Science Protection Act, and you have to get special qualifications to be able to keep them and look after them and then do experiments on their tissues. But in the States, mice don't even count as animals that need to be regulated in science. So different experiments take place. Like anything that I did to the mice had to be completely justified and overseen by an ethics committee. So I could square working with animals, well, with working with mice in the UK. Can you edit your genetic immune system to fight off a foreign gene specifically in a particular place in your body? So, I mean, that's a big question in science, because if we can do so, then we can potentially give people organ transplants and not have to give them immunosuppressants for the rest of their life. Hard, very hard. What would you do? So some people who are less comfortable with it. Okay, so no one likes putting down, or none of the researchers I worked with liked putting down or sacrificing the mice that we used in experiments. None of us did. So asking someone else to do all of your putting animals down for you was seen as quite rude, because it's not really a fun way to spend your day at all. You can do some things in cell culture, but realistically, especially if you want to scale up into stuff that is applicable to humans, you're going to need animal models at some point. So you would probably collaborate with people that worked with animal models, but then ethically, is that different? I don't know. Can the phenotypic effects of gene editing be accurately predicted? Again, big question. In theory, if you're specific and you have no off-target effects, yes, but that's a big if. What kind of precautions are in place? Okay, so we're going to come on to that now. So problem with CRISPR is that because it's cheap, and I know that some of you are not as convinced that as I am that it is cheap. Because it is cheap, just trust me on that, and relatively easy to use, you can't just be like, oh, in the future, there's a hypothetical person who could genetically engineer babies. Like, actually, that has happened now. So I don't know if any of you know about this, but it's a very interesting thing to look up and read about. So in late 2018, a scientist claimed to have used CRISPR to disable a gene called CCR5 in twin girls. Does anyone know what CCR5 does? I'm expecting no, but if you want to Google and then just tell me in the chat, I'm not. Oh, Iman, oh my goodness. Okay. I don't have a favorite participant, but you're near the top of the list is all I'm going to say. Yes, it was to prevent HIV infection was his rationale of why he removed this gene. Is that a good rationale? Well, so people who are HIV positive in a country that gives them access to antiretroviral therapy actually have an increased life expectancy compared to people who aren't HIV positive now, probably because they're surveyed more carefully and like kept an eye on. But so is stopping someone from getting HIV then actually a good justification of doing something that could be incredibly risky? Like, he claims there are no off-target effects. Those twin girls won't know until they're like 60. What if they get early Alzheimer's? What if they get leukemia when they turn 25? Yeah, I mean, I don't think so either. And also the majority of the scientific community did not think so. It was appalling. Thank you, Remy. It's very interesting to read about, and it's very interesting to read about the repercussions. Their autonomy is taken away is a really good point, because they were babies, exactly. So they can't consent. So if they do grow up and then get leukemia at 30, that's kind of rubbish and not really fair at all. So really interesting things to think about, especially because it's not like this is a hypothetical anymore. This is happening. And I think the thing that this really made people in science think was we need to actually get some consensus and regulation in this field. We can't just kind of have a gentleman's agreement that no one edits the babies, because people are doing that. Yes, there was parental consent, but is that sufficient in this situation? It's a really interesting ethical debate, and one that we don't really have like right answers to. It's very much like an area of research. The other thing that I just wanted to, and I'm very near the end of my talk now, is this condition, Leber's congenital amaurosis, which is all about blindness. So on the screen in front of you are two images of retinas. So this is what, like when a doctor gets the little black thing on a stick and goes up to your eye like this is what they see at the back of your retina through your pupil. So hazard a guess, anyone, which one of these do you think is normal? The right one or the left one? The right one, the left one, the right one, the right one, the right one. It is, the right one is normal. I'm very sorry. So I won't say your name, the person who guessed the left one, because to be fair, the person I tried this talk on did also guess left. So those of you who said the right one is normal, could you have a go at describing what is different on the left one? What made you think that the left one was abnormal? It's dust. Okay, I see what you mean about being dusty. The blood vessels aren't clear. Okay, more cloudier. Right, what I'm going to draw your attention to is all these squiggles around the middle. So what you've got there is what we call neovascularization. You have more blood vessel growth, and that's blocking off the optic disc and leads to blindness in childhood or early adulthood. Now, all you need to know is that the eye is immunologically privileged. You don't really need to understand the whys and wherefores of that, but essentially, you know how we've mentioned like the immune system reacting to foreign things and rejecting organs. I know you guys have had a talk on that already. The eye doesn't really get that. So it's a really good target for gene editing, because if we introduce a protein that the body thinks is foreign, it's less likely to lead to a massive immune response, okay? So this is actually like editing the gene that causes, or one of the genes, there's like 20 odd of them, that causes this condition using CRISPR has actually been done. Gene editing's been done in eyes, certainly, for a while, using viruses, kind of like the virus vector that was used for the Oxford COVID vaccine. So using a virus to introduce some kind of RNA into a cell. But for this one, this particular gene, they used CRISPR, and that's because the gene itself was so long that a virus vector couldn't fit it in. Like most of them can't carry much DNA, okay? So this is another really up and coming, like I picked two research papers that were really recent, because I thought you guys deserved that kind of stuff. And I can't go into them in too much detail because I'm not going to lie, they're quite far above the A-level syllabus. But if you're interested, then go away and have a Google, because there's some really good popular science write-ups. And the more of those you read, the more likely you are to be able to understand like the detailed ones. Now, we're not going to have time to do this today. I'll just go to the question slide. But what I was going to get you to do was to have a think about how you, if you were a researcher that had all the money and time in the world, what experiment would you design? Like, is there a disease that you're interested in? Is there a tissue where you know that disease is active? What would you do? Would you turn on the gene, turn it off, visualize it, do something else maybe? You might need to do some extra reading on that. How could you work out the function of the gene in question in terms of the cell, in terms of the DNA within the cell, or like subcellular things, or even in terms of like the tissue system? So something for you to go away and think about if there's a particular disease or gene that you're interested in, and maybe just use that as a springboard to doing some more reading, some more thinking, watching YouTube videos, whatever it is, and just taking your knowledge to that like superstar level where you're going to be able to write a smashing personal statement and get into medical school. So final opportunity for questions. Thank you very much for all of your amazing participation today. And please, please, please do the feedback. Remy's just put it in the chat and there's a QR code as well. And I will answer the last few questions. Can gene editing be used to regenerate cells, or is this more of a stem cell thing? So the thing that you want to look up, Adriana, are induced pluripotent stem cells. So I have a like genetics workshop that I give sometimes to students around your age, where we use the two technologies to do that disease designing, like experiment designing thing. So look up the iPSCs, again, recent technology within the last 15 years, I think. Again, recent Nobel Prize winning science. So that's the thing that you want to look up if you're saying regenerating stem cells. In theory, yes, we can regenerate stem cells from any tissue in any organism. Where do you think gene editing will go next? Oh gosh, I think that it will move into more of the therapeutic side of things. I think it will be really cool to see how it goes in terms of like bone marrow transplants and also in terms of cancer. I think there's probably quite a wide, like I didn't do much cancer science in my DPhil, so I only really know about it from like listening to other people's talks. But I think that there's really, really interesting personalized medicine stuff on cancer that I think Holly's giving you guys a talk on in a couple of weeks. You're welcome, Eman and Adriana. Thank you very much. What are good papers to read for an interest gene editing? So I'm just going to look at Remy's face and say, can I find some good papers and then send? Can you send them round?
Yeah, of course. When I send the email up for the next talk, I'll include your recommendations.
Okay, cool. I'll try and come up with like 2 or 3 like good primers on gene editing. Can we use gene editing on certain areas of the brain to control certain functions as a result? Okay, so bro, what you want to look up is optogenetics. That you want to Google that and have a look. So it's not really gene editing, it's more you use gene editing to introduce light sensors into neurons, and then when you shine a light on the neuron turns on. So that's what you want to look up. It's really, really cool neurology. I'm currently doing my GCSEs and hope to do sciences for A-level. Is there a specific career path to become a genetic engineer? I'd say study some kind of life science at university, so that could be biology or chemistry, but it's up to you. Have a look at different university prospectuses. If you start doing that now, then you can look at like what modules different universities offer and like whether they offer like a year in industry or a year doing research and things like that, because that's probably what you'd be interested in. I'm just guessing that would be like how you would go down that pathway. But to be honest, I'm going to put molecular biology because that's like the kind of science you want. Molecular biology, that's the kind of science that you want to do if you want to do gene editing. Okay, so if you see that, be like, aha, that's me. You're all welcome, guys. Thank you for the feedback. I'm glad you've enjoyed it. It can, but the difficulty is you need to edit more cells. And often, like, you know, the embryo doesn't have an immune system. So if you introduce something foreign into like a 2 or 4 cell embryo, it doesn't really know it's foreign. If you introduce, like, if I inhaled a CRISPR-Cas9 aerosol that was meant to like remove the cystic fibrosis gene, then my immune system would be like, what is this? No. And like, that's been the problem with gene therapy the whole way through, is as soon as you introduce foreign DNA, you're normally okay the first time your body sees it. The second time, your body's immune system is like, absolutely not, and goes into overdrive because it thinks you're being infected. So in theory, Yes, on selected tissues. In theory, yes, if we like manage to dampen down the immune system. In theory, yes, if you maybe take the tissue, like the bone marrow, that's one that people have talked about, out of the body, change it, put it back in. But there are lots of caveats. So an area to read into, but like an area of active research. Okay, are there any more questions? Could you stimulate stem cell differentiation? Yes, absolutely. Look at your iPSCs, so your induced pluripotent stem cells. And yeah, the current science, or like our current capacity, is like, I could take some skin cells from like a person with Parkinson's, re-differentiate them into stem cells, like undifferentiate them, I guess, into stem cells, and then re-differentiate them into brain cells, just by adding different things and growing them in different medium. And then I'd wind up with a load of brain cells with whatever genetics that person with Parkinson's has, which is obviously pretty great if you want to study it, because like taking a brain biopsy from someone is kind of frowned upon, because think about it, guys, like it's not really good. So yes, you can stimulate stem cell differentiation, and you can also undifferentiate adult cells. If you don't mind, what's the current project you're working on? Okay, so this is a lot more clinical. The most recent thing I had published was about people diagnosed with cystic fibrosis in adults. There are some of them, they have particular kind of types of mutations, and I was looking at like how they tend to present and how we find them. The thing I'm working on now is like a little research side project, is looking at pediatric respiratory presentations. This is quite boring, guys, I'm really sorry, to emergency departments this winter and last winter. So normally kids get really snotty in the winter. If you've got younger siblings, you may have noticed this. And they get loads of respiratory diseases, and pediatric departments are super busy. Because of all the social distancing this year, that didn't happen. And people kind of know that anecdotally, but me and one of my friends were like, it'd be quite cool if we could quantify this and kind of show a translation into bed occupancy and show like which diseases were particularly affected. So we're doing the data collection for that now, which has taken quite a long time and been quite boring. But when we've got that done, then it won't be too much work to pull together the figures and then write the paper. So that's what I'm doing now. It's probably not very interesting, but there you go. What are your other favorites in gene editing? What am I interested about in gene editing? Okay, so I said this before to Remy and Jade. So this isn't really gene editing, but it's very much genetics linked. So one of the cool things, I think, at the moment is using really small molecules as therapeutics. So there are lots of like libraries of small molecules that are just like random chemicals that drug companies have. And if you do screens with those, you can often find that they treat particular conditions. So for example, you could get a small molecule that alters how genes are expressed that would lead to muscular dystrophy being cured, which is literally a treatment that's come through in the past few years. And I think they're really cool because they tend to be a lot cheaper than genetic therapies. They don't tend to have rejection issues, and they're also scalable, as in like they're a lot easier to spread around the globe than genetic therapies, where you tend to only get access to them in like really specialized hospitals. Could neurodegenerative diseases like dementia be treated through gene editing? So I think the potential that if we're reaching up to the sky and like really waving our hands around in the air, we know that neurodegeneration means that like neurons die off. So potentially if we could undifferentiate and then redifferentiate someone's skin cells to be neurons and then inject them back into someone's brain, and maybe have like cleaned them up in the meantime with gene editing, that might be something. But to be honest, I think the small molecule thing is going to be better. But I think this is also the really cool thing about science, is we literally have no idea where the next kind of brainwave will come through. No one even realized that CRISPR-Cas9 was a thing until like 10 years ago. People didn't realize it was a thing, and now it's completely transformed how we do science. So we don't know. I don't know if you could treat them through gene editing that late in life, because when the damage has taken place, you kind of need to replace the cells somehow.